Is it on? Yeah. Okay. Welcome, everybody. My name is Klaus-Michael Weltring, and I will chair this session. And I have to admit that I don't understand a lot, at least from the first three talks, so that's why I'm eager to learn more about these data-producing techniques, which we will hear about. The session is called Technologies Enabling Personalized Medicine, and as you might be well aware, one thing is, of course, to have enough data to make personalization possible. Then you need tools to really address the personal treatment, diagnosis, and so on. So to all of these aspects, we will have talks today. The first talks will deal with the data to be collected and managed. And the first talk in this session is by Professor Dr. Renato Paro from Basel University. So we start with a local scientist and expert. And he will give an overview of analytical tools for clinic deep sequencing. And this is certainly a very hot topic at the moment. So I'm looking forward to your talk. Thank you for coming.
Thank you for the... Can you switch, please? Okay. Thank you for the introduction, and also thank you to the organizers for inviting me. Now, indeed, actually, I was prepared to explain to the audience why I am a local, since I am a professor at ETH Zurich. And so everybody asked me, what are you doing in Basel? Indeed, it turns out that ETH Zurich has a department here in Basel. It's a department of Biosystem Science and Engineering. Now, as we all know, going from the genotype to the phenotype is one of the major questions in biology. It's clear that there is no direct connection from the genotype, which is arguably the most stable information that we can get in living cells, that we can analyze, that we can quantitate, that we can sequence. Okay? But going from the sequence to the phenotype is essentially made difficult by all the intervening filters, namely not every piece of DNA is transcribed, not every transcript is translated into a protein, and so on and so on. All these filters we need to understand in order to be able to deduce the information that we gather from the genotype into a corresponding phenotype. So one filter that we will be discussing here today extensively is the first filter controlling gene expression, epigenetic control of DNA information. But one should not forget that there are additional ones, like quality control mechanism, chaperoning, for example. So even if a protein is produced, it not necessarily means that it automatically is involved in a process, because there is a quality control that checks out whether the protein is folded appropriately. Physiological processes contribute to this filtering of genetic information. Okay? Now, as a molecular biologist, as somebody involved in basic science, we know a lot about this part here. The clinician, the doctor, sees mostly the phenotype and would like to understand where that particular disease phenotype, for example, comes from, whether it has an underlying genetic cause. Now, one way of trying to connect genotype to phenotype is utilizing the new technologies that allow us to read the genotype much faster and more efficiently. The whole field is called genomics. We know that a genome is a sum of all the individual, all the genes of an individual, and thus genomics is the study of all the genes of a cell or tissue at the DNA called genotype, at the messenger RNA transcriptome, or at the protein level, at the proteome. And these are several different names for these new technologies that allow us to read sequence much more efficiently. So it started about 2005, and by 2009, several technologies were available on the market. You see some of them, some have already disappeared, and by now we have these 4 technologies that are on the market and substantially used by the sequencing centers or the laboratories utilizing genome information. Okay? The 454 system from Roche, the Illumina system, which is mostly the workhorse of most sequencing laboratories, it turns out, and as well newer technologies from Ion Torrent and from Pacific Bioscience. So these technologies have advantages and disadvantages depending on what your needs are. You might use one or the other. And this is just a compiling of the advantages that these three technologies have that are currently being used most in the sequencing centers from PacBio, Ion Torrent, and as mentioned before from the workhorse Illumina. You can see, for example, with PacBio, you produce relatively long reads. Okay? This is an advantage, for example, if your read contains repetitive sequences, long reads can span short repetitive sequences, allowing also to map these sequences on the genome. Okay? Conversely, the Illumina technology produces relatively short reads, but as you can see here, it produces a lot of short reads, and hence you can utilize those using statistical method, bioinformatics method, to map these reads on a reference genome. Okay? There are other advantages that are listed here, and I won't go into the detail, but for example, the PacBio system allows you even to identify methylated Cs and modified bases, which are an important issue when you want to study epigenetic control mechanisms. So very briefly, let me just explain to you two technologies. Obviously, I will not be able to do this in all the detail that would be required to really understand the system, but Illumina, you produce short DNA fragments from your library. These short DNA fragments are, you add linkers, which are utilized to anchor these fragments on a particular surface. And then these fragments are amplified using the PCR reaction, and ultimately you end up with these clusters of identical DNA that all derive from the same original fragment that was localized. And essentially the Illumina sequencer is just a fancy microscope, because what turns out is then that you do the regular Sanger sequencing reaction using fluorescent nucleotides, and with a big microscope, since you have a lot of DNA on one cluster that you can visualize under a microscope, you can then read, so to speak, the addition of every nucleotide that that the reaction produces, the addition of every nucleotide, which is differently labeled with different fluorochromes. And so over time, you synthesize the strand, the new strand, and you can, during the synthesis, read every new nucleotide and deduce the sequence of that particular short nucleotide. Another technology that is becoming more and more utilized is the PacBio, Pacific Bioscience real-time sequencing technology. This uses a totally different principle, namely it uses the enzymatic activity of the DNA polymerase, which is anchored to a particular surface, and the DNA passes through this polymerase, and one strand is then synthesized by the polymerase, and the incorporation of a fluorescent nucleotide is measured in this well here, and every nucleotide that is added will give a different signal, and hence the system can record real time the synthesis of the nucleotide, and hence determine the signals in real time. And the advantage of this, as mentioned before, is that you can sequence relatively long strands with the possibility to overcome, for example, short repeats. So what are the application of some of these technologies? Okay? Well, first of all, we have a reference genome we have a human reference genome, and we can now start to map aberrant genomes like cancer genomes in comparison to this reference genome. We can map polymorphisms, and we can map other things that are found different in different individuals in their genome. But we can also sequence the transcripts. We can sequence the messenger RNAs. As I will show you later, there is a method where we can even identify the binding of particular transcription factors or chromatin proteins at their target sequence, at their target gene. So using this new technology, there are different consortia, like the International Cancer Genome Consortium, that is trying to identify the genetic aberrations correlated with particular cancer genomes. And you probably realize that there is a massive increase in publications finding, sequencing cancer genomes and finding corresponding cancer genes, driver genes in these cancer genomes. But there is also the project of 1,000 genomes which tries to utilize this technique in order to determine human genetic variations, okay, that all make us somehow different. These are the SNPs and smaller changes in the DNA sequence that are also important for pharmaceutical research, for example, because it's very clear that our genome, to a certain degree, contributes to the way that we react on certain treatments by drugs, for example. And so ideally, we would want to know our own genome in order eventually to be able to allow to predict how we react to certain drugs on long term. However, additionally, you can, and this is, for example, part of our work, we mapped these genetic variations in certain cancers. We can, for example, concordant reads means these are the same DNA sequences as in the reference genome, okay? And these will be the aberrations that we observe in a cancer, corresponding cancer genome. We can even discriminate between homozygous and heterozygous variations. But what has also to be clear, because so many sequences are produced, it needs a dedicated bioinformatics support to make sense of all this information. And a lot of people who have great ideas about sequencing genomes or transcripts or whatever underestimate the requirement and the effort it takes to bioinformatically analyze the sequences that come out of these machines and make sense out of it. Okay? So, for example, we needed to develop a new algorithm which we call deterministic structural variation detection using so-called long overlapping paired end reads from these sequencing machines. And incidentally, I won't go into the detail, but it turns out that in this particular cancer that we studied, there are no genetic aberrations, but it's simply driven by epigenetic changes in that cancer genome. Okay? So these are novel things that one discovers by looking in detail at these tumors. Another aspect is we can now really down to the single exon map the different splicing forms that a cell produces, splice forms of the transcript. Okay? Also this requiring extensive bioinformatics support because it's very difficult to combine the sequences in an appropriate manner. But for example, you can also use this technique, in this case it's called PAR-clip, for mapping RNA-protein interactions, mapping proteins that are bound to particular transcripts, non-coding RNAs or messenger RNAs, and we can utilize this PAR-clip method to specifically identify the sites on the transcript that interact with a particular protein. Okay? We can use the so-called chromatin immunoprecipitation methodology that allows us to map not only the sequence of the DNA, but also to map those proteins and histone modifications, for example, that are bound to that particular sequence. Okay? And on top of that, we can even map these proteins and interacting DNA domains that are responsible for producing a particular three-dimensional domain in the nucleus and contribute to the regulation of this nucleus. And so at the end, what we have is, we are very good at that, to produce a lot of information that covers how this filter works from the initial DNA sequence to the gene regulatory to the epigenetic mechanisms. We still lack information from this side, but the advance of the new technology certainly allowed us to develop a lot of information at this end of the big question that we have from genotype to phenotype. And with this, I would like to end. Thanks a lot for your attention. Questions at the end. Fragen am Schluss. Okay.
Are there any questions?
So concerning your first question, I hope I made it clear that no, the sequence does not necessarily tell you immediately what the phenotype will look like. Because indeed, this is, as I mentioned, I call them filters, but there are many intervening regulatory systems that a cell utilizes to interpret that sequence. Okay? And this can vary from cell to cell, from environmental condition to environmental conditions. And this is what we need to understand first before we can simply deduce from the sequence how the phenotype will look like. Okay? For one phenotype, it will be easier than for the other. Okay? But even simple things like the height of persons, we cannot get a one-to-one ratio from the sequence to the phenotype. What concerns your question about technology, the 454, I wasn't quite sure what you meant by. I think it very much depends what your needs are. There are laboratories who need 454, but if you look how the market responds to these technologies, it's clear that like Illumina is the leader. Okay? It produces a lot of short reads, and we have bioinformatic tools to utilize even these short reads in a comprehensive manner to interpret the genetic information. And it's much faster than other technologies. Okay? So this is why it has been so successful.
Eva Stępień from Poland. I have a rather common question. We use NGS for diagnostic of cancer patients in our hospital, and good laboratory practice recommends that every NGS mutation must be confirmed by Sandra by traditional sequence.
By PCR.
By PCR or by Sandra.
By classical Sanger sequence.
Yeah,
yes.
because of there is a lot of errors in this method still, and there are some chips which are dedicated to sequence the most common mutations in cancer. We don't need sequence all genome of every patient to diagnose mutation in cancer patients.
Right. So, indeed, if you want to do it right, you would need to sequence very deep these cancer genomes. Okay? But one major problem with cancers is also that the homogeneity or the heterogeneity of the material that is used for the sequencing to produce the libraries for the sequences. So there, it's very difficult to assess whether you have one driver mutation, for example, that is characteristic for that cancer, or whether it's just one of these passenger mutations. And hence, to get all this information, you need to sequence very, very deep, meaning you have to have a lot of reads. Okay? The question is, can this be really applied in the clinic as a diagnostic tool yet? Okay? And I'm still not convinced that, yes, of course, you can footprint the genome of that particular cancer type of that particular patient. But I'm not really convinced that this can be used then in the subsequent treatment of that cancer patient to the degree that you would like it to do.
Some time for questions, general questions at the end of the session. Now, thank you very much, first of all, again.